Bismillahirrahmanirrahim. 9700 June 24 paper 2-3, the first video, which will do the first 2 or 3 questions. As you all know, this paper is for 1 hour 15 minutes, and you must try and finish it well in time so as to be able to revise it once before you submit the paper. Bismillahirrahmanirrahim. Starting with question number 1, the Zika virus is a pathogen that can infect human cells. Pathogen, disease causing microorganism. Figure 1.1 is drawing of a structure of a Zika virus. So they've told you this is a virus, it's not a bacteria, it's a virus. In figure 1.1, structure A is made of protein. So we're talking of structure A, which is inside, and then we have something inside it. So name structure A. What is it? It's a capsid. Right? You could have said capsomere, you could have said a protein coat as well. That was also acceptable by Cambridge. Then it says in figure 1.1, structure B is a single-stranded molecule. Suggest the name of structure B. It could be anything. It could be RNA or DNA. That is what you have studied in A2, that it could have been DNA or RNA. Actually, it's RNA because it's a retrovirus if you're going to talk about HIV. So you could have both the DNA and the RNA can be double-stranded or single-stranded, and that's of course a part of the A2 syllabus, which if you're doing the AS you won't know, but of course I always tell you that. Figure 1.2 is a transmission electron micrograph of human kidney cells infected with Zika viruses, and magnification is 200,000 times. Calculate the actual diameter of a Zika virus using the line XY. So you have to measure that line and give your answer in nanometers. So the unit is given here, nanometers. So you measure this, and this was how much was it? So measuring this XY, it was 1 centimeter. So I always do it like this: 1 centimeter is 10 millimeter. And then I make it a point to underline the 3 zeros because 1 millimeter is equal to 1000 micrometer. So 10 will be 10,000. And then, 3 zeros for the micrometer and 3 zeros for the nanometers. And then you divide it by the magnification, which was here. And this is the answer that you get. But you always make a careless mistake. I call careless mistakes now CM. So Gen Z is very good in making careless mistakes. You are brilliant in making careless mistakes. So please avoid the careless mistakes because this is going to cost you a grade. You might be brilliant in making careless mistakes, but you must be a chunker then. Then coming to part 2, magnification of 200,000 cannot be achieved by a light microscope. The maximum useful magnification is x 1500. I said maximum useful magnification. So useful magnification means you can magnify more, but it will be at blurred. So you're not going to see it any more clearer, or you're not going to see any more details. Clear also is a word which you cannot use. So a magnification of 200,000 cannot be achieved by a light microscope. The resolution of a transmission electron microscope is also higher than a light microscope. Describe what is meant by the resolution of a microscope. Ability to distinguish between 2 points. This is how you would write it. You must know these wordings and you must sort of memorize these. Some of the things you have to rote learn. So this is one of the things that you need to be really actually memorizing or rote learning. Now coming to part C of the question, the vector for Zika virus is the mosquito Aedes aegypti. The mosquito feeds on the blood of an infected person, transmits the virus to another person when it feeds again. So the vector for Zika virus is the mosquito Aedes. The mosquito feeds on the blood of an infected person, transmits the virus to another person when it feeds again. Describe the similarities and differences between the transmission of Zika virus disease and the transmission of malaria. So transmission of malaria, similarities and difference. So malaria is transmitted by Anopheles rather than Aedes. And then both are transmitted by vectors, both are transmitted by mosquitoes, both are blood-borne diseases. That means the vectors feed on the blood. So malaria by Anopheles rather than Aedes, that's the difference. That is the only difference that we have. And the similarities are both are transmitted by vectors, both are transmitted by mosquitoes, and both are blood-borne diseases. Now coming to part D of the question, Zika virus vaccines have been developed by scientists. One of the vaccines contains small proteins from the Zika virus. So the virus will have some proteins on its surface, which are antigens. So one of the vaccines contains small proteins, so they can just be giving this thing and that will trigger an immune response. So these are called antigens. Explain how giving this vaccine to a person can lead to the development of long-term immunity against Zika virus disease. Now, it's very simple. Proteins are foreign antigens or they're non-self. Proteins stimulate a primary immune response. B and T lymphocytes are activated. It results in clonal expansion, which means cells divide by mitosis to produce a clone. And then formation of memory cells, whether it's the B lymphocytes or the T lymphocytes. This all you get one mark for that. You could have said, what is this called? This is called artificial active immunity. Then the memory cells remain in the circulation for a very long time. And then if this virus enters, then there is a secondary immune response is much stronger and faster production of specific antibodies. So the person does not have the symptoms of the disease when infected with the actual Zika virus. So all these points, there were a lot of points, and you could have only given me four of these to get the 4 out of 4 for this question. So a lot of points, and you could have given me any four of these. Proteins are foreign antigens. Antigens stimulate immune response. T lymphocyte or B lymphocytes are binding to the antigen and cause activation, then clonal expansion, then formation of memory cells, and this is artificial active immunity. Then coming to the last part of this question, explain how a vaccination program may limit the spread of the Zika virus disease through a population, through a group of people living in a city or a town or a country even. So this is going to result in herd immunity. Now, what does the word herd immunity means? It means a large proportion of the population is immune. They will not suffer from the disease. Immune means they will not suffer from the disease. So people who are unvaccinated, unlikely to be in contact with an infected person. Or you could have said higher, we can achieve a higher percentage cover, greater percentage of the population need vaccinating if you say this is not going to work. So the word herd immunity, if you knew that, well, that's a very interesting word, and I'm sure you must have read about it, and if you had focused, you probably understand it as well. But what does it mean? It means a large problem. If you've vaccinated a lot of people, in the 90% of the population, then 85% of the people are immune. If you've only vaccinated 50%, then 45% are immune. So because everybody doesn't get the vaccination doesn't work on everybody. So herd immunity is a large proportion of the population is immune. So they've been vaccinated. People without immunity unlikely to be in contact with an infected person. So if 10% of the people have not been vaccinated, then they have less chances of meeting a person who is an infected case. Question number 2. Collagen is a fibrous protein that is found in many tissues in animals. Describe the structure of a collagen molecule and structure of a collagen fiber. Now I need to show you some diagrams before we go on to this question. Now here's a diagram showing you collagen. Now you can see here, these are the amino acid chains, and every third amino acid is glycine. You can see here, every third amino acid is glycine. And then this is twisted around each other. Three chains are twisted around each other. And these three chains, when they're twisted around each other, becomes a collagen fibril. And many of these fibrils together is known as a collagen fiber. I'm going to show you another diagram. Now, in this diagram, you can see here peptide chains. So the amino acids link together to form these three peptide chains, and the three peptide chains then twist around each other and then form a collagen fibril and then collagen fibers. So now that you've understood this, you can do this question very easily. So now you can look at the points. Of course, they were very similar to what I just explained to you. Three polypeptides, helical chains tightly wound. Every third amino acid is glycine, hydrogen bonds between polypeptides. Now the collagen fiber is molecules arranged in parallel manner. Covalent bonds between molecules and molecules in the fiber have staggered ends. Now coming to the B part of the question, some amino acids in collagen can be modified to improve the stability of the protein. For example, the amino acid lysine can be modified from hydroxylysine. Figure 2.1 shows a disaccharide bond to the amino acid. A disaccharide bonded to the amino acid hydroxylysine in a collagen molecule. The disaccharide is made from the monosaccharides, which are indicated by the labels D and E. So these are two monosaccharides and they are joined together and they have been bonded to the amino acid hydroxylysine in a collagen molecule. And D and E are two monosaccharides. Right? Everybody's clear on this diagram? Now let's see what are they asking. Give the precise name of the monosaccharide labeled D. Now what is D? Let's look at D. D is an alpha glucose. Why? What was the rule that we followed? Alpha was below, the OH was below. So this is carbon 1. So the OH is below. Alpha below, beta above. That's why the mnemonic is ABA, A, B, B, A. So this is an alpha glucose. So you would say alpha glucose. Then it says on figure 2.1, label the glycosidic bond with the letter G. So this would be the glycosidic bond. This would be the glycosidic bond between, of course, it's not 1 and 4 or 1 and 6, it's 1 and 2. So this would be the glycosidic bond. Then it says on figure 2.1, draw a circle around the R group of hydroxylysine and label the circle with the letter R. Now it says draw a circle around the R group of hydroxylysine and label the circle with the letter R. Now let's do that. So this would be the R group. It's always on the central carbon, NCC, central carbon. And this would be the R group, so we label it R. Write your answer on figure 2.1. Okay, so that finishes this part of the question. Now let's go on to, it says osteogenesis imperfecta is a disease that results from a deficiency in collagen. Suggest how a named tissue or structure is affected in a person who has this disease. Where is collagen found? Collagen is found in the wall of the arteries, in blood vessels, in cartilage, in the bone, in ligaments, in the skin, in tendons. So if it's not deposited in the bones and the skin and the tendons, they're going to be weaker, deformed, easily broken. The blood vessels or the cartilage, let's talk about the bone. Bone will be weaker, it will be easily deformed or easily broken. So basically you have to know where collagen was found and if it was affected. Like if it was the skin, then the skin would be all very wrinkled, like it happens in old age, collagen is lost and skin becomes wrinkled. So in any way, if you give me a description of how the function would be affected, if you said blood vessels, and then you would have said, okay, well, they will be thin, the walls will not have enough collagen, so they will be weaker. They won't have that strength to withstand the high pressure of blood in the arteries. So any way in which you showed the effect, how the function was affected, you would get that mark for that. Now coming to question number 3. Arteries, capillaries, and veins are 3 types of blood vessels. Table 3.1 shows some features of these 3 types of blood vessels. Complete table 3.1 by using a tick if the feature is present in the blood vessel and a cross if the feature is absent from the blood vessel. Put a tick or a cross in every box. And some of you don't do that. Gen Z, careless mistakes. Okay, smooth muscles. Smooth muscles are present in artery, no smooth muscle in capillary, present in vein. Then endothelium is present in all 3. Endothelium is the lining. Of course, capillary only has the endothelium. Then tunica media is present here, not in a capillary, and here as well. So 3 marks, this would be per row. If you get all these rows correct, if the entire line is correct, then you get 1 mark. If this line is correct, then you get 1 mark. So please remember that these are marked per line. Then we come on to the B part of the question. Figure 3.1 shows the transmission electron micrograph of part of an alveolus and part of the adjacent capillary. So they've shown you an alveolar airspace and they've showed you a capillary lumen. Draw 2 labeled arrows on figure 3.1 to show the direction of movement of oxygen and the direction of movement of carbon dioxide during gas exchange in the lungs. Very basic question. So the arrows, this was only one mark. Naturally, oxygen is going to go from the alveolar airspace into the capillary, into the blood, and carbon dioxide is going to be diffusing the other way around. So oxygen will leaving the alveolar space, entering the capillary, or carbon dioxide will be leaving the capillary lumen and entering the alveolar space. Then part two of the question, suggest and explain how a steep oxygen concentration gradient is maintained in the lungs. Basically, blood arriving in the alveolus is deoxygenated. Oxygen taken up by hemoglobin. Newly oxygenated blood is constantly removed. A large network of capillaries. Then ventilation, which is inhalation, and exhalation allows more oxygen into the alveolar space, or you can say air is refreshed. And ventilation maintains a large concentration difference between the alveolar space and the capillary. Basically, what to understand that this blood entering here in the lungs is deoxygenated. So oxygen is more here inside the alveoli, so that is going to diffuse in. But then this blood, as soon as it gets oxygenated, is flowing. The continuous flow of blood is going to ensure that as soon as this deoxygenated blood gets oxygen, it's carried away. So it's carried away, and then deoxygenated comes from more of it comes out here. So deoxygenated, as soon as it gets oxygenated, it moves on, and then deoxygenated comes more. So then deoxygenated is here, oxygen is more in the alveoli, so oxygen is going to diffuse in. So basically, how are we going to word it? Blood arriving is deoxygenated, then formation of oxyhemoglobin, then this oxygenated blood is continuously removed or continually removed. Large network of alveolar capillaries which is going to carry away this blood, bring and carry away the blood, and inhalation and exhalation will constantly replace the alveolar air. So if there's going to be more carbon dioxide in the alveolar air, even that is not good. So that needs to be taken out. The carbon dioxide must be exhaled out. And when you inhale, then the inhaled air will contain more oxygen in it and less carbon dioxide. Part C, the passage outlines the roles of blood vessels associated with the heart. Complete the passage by using the most appropriate scientific terms. The dash carries blood to the left atrium, pulmonary vein. So the pulmonary vein carries blood to the left atrium. After passing from the left atrium to the left ventricle, blood is pumped into the aorta. The aorta is one of the two large arteries that carry blood away from the ventricles of the heart. Blood that leaves the heart to enter these arteries must pass through the semilunar valves. Semilunar valves. Oxygenated blood is supplied to the cardiac muscle cells through the coronary arteries. So an easy 3 marks, and the total marks for this question were 11. Question number 4. Lignin and suberin are polymers that are present in plant tissues. Describe and explain the roles of lignin and suberin in the transport of water through the roots and stem of a plant. So basically, lignin and suberin are is hydrophobic, impermeable to water. So it's a waterproof barrier. Then suberin is found in the Casparian strip because it says describe and explain. So you must tell me where they are found. Suberin is found in the Casparian strip. You can see the endodermal cells, or you could have even said endodermis. So water must move from the apoplast to the symplast. So this allows the control of solutes before entering the endodermal cells or the xylem. Now, lignin is found in the cell walls of xylem. So you could have given both different headings and talked about them. Lignin is found in the cell walls of the xylem vessel elements. You could have said xylem, you could have said vessel elements. Lignin prevents water leaving the xylem. Then lignified walls prevent the collapse. Please do not use the word bursting. Collapse is something else and bursting is something else. Then the lignin provides strength for the structural support of the xylem. So all these points were available for you. There are about 7.8 points which were available, and any you had to give me two for lignin and two for suberin. So you couldn't have given me all the points for just one and get the 4 marks. We would be asked as examiners to maximum give you 3 marks if you gave just suberin or just lignin. So wording it, so generally there was a statement lignin and suberin impermeable to water. That's why I've written that in green. Then the red ones are for the suberin. Suberin found in the Casparian strips. Water moves from apoplast to symplast, allows control of solutes before entering the xylem. Then lignin found in the wall of the xylem vessel elements and lignified walls prevent water leaving the xylem. That's why they have the pits, which allows water to move out. Then lignified walls prevent collapse of the xylem and provide strength for support. Part B says the enzyme lactase catalyzes the formation of lignin in plants. Lactase. Figure 4.1 is a diagram of the mode of action of lactase. So we see the enzyme lactase, which has copper ions in its active site, and then monolignols sort of attach there, and then they're two separate, then start of the lignin chain. Very interesting, figure 4.1. And then it says describe and explain the mode of action of lactase when catalyzing the formation of lignin. Let's look at it again and then let's concentrate on it. So basically, what have you to give me? It's a very simple thing. It says lactase uses induced fit. Why did we use the word induced fit? Now look what is happening. Now this is the active site, and then you see the active site changes shape, and the word that I use often is molds around the substrate, and then it goes back to its shape once the if it is catalyzed the reaction. So lactase uses induced fit. Active site shape not fully complementary to shape of monolignols. Then active site changes shape to fit monolignols. Then becomes fully complementary, forming enzyme substrate complex, lowers the activation energy, and lactase returns to original shape after the product leaves the active site. So basically, you have to look at the diagram very carefully and give me all these points, which were very, very relevant and very easy to look at if you had looked at it a little carefully. Now, writing it, wording it, lactase uses induced fit, active site shape not fully complementary to shape of monolignols, which is the substrate. Active site changes shape to fit to the monolignols, becomes fully complementary. Complementary is underlined, so you have to use this word. You can't use any other alternative words. Forming enzyme substrate complex, lowers activation energy, lactase returns to original shape after product leaves the active site. Now, whenever you're reading this sort of a question, remember it says, whenever it says on the diagram, it says figure 4.1 is a diagram of the mode of action of lactase, and then the question is based on figure 4.1. So that is why I describe and explain when I catalyzing the formation of lignin. Question 1, 2, 3, and 4 have been answered in this first video on this paper.